Welcome to The Explainer. Okay, imagine for a second that you have to create a library for every single living thing on Earth. I mean, where would you even begin? Well, today, we're diving into the brilliant system scientists developed to bring order to the beautiful chaos of life. And we're going to meet the tiny pioneers that started it all.
So let's just start with the scale of the problem. Scientists figure there are something like 8.7 million different species on our planet. That is just a staggering number, right? With that much diversity, how in the world do you even start to make sense of it all?
You can really think of it like trying to organize a colossal planet-sized library. Now, the first attempts to catalog life, well, they created some pretty confusing shelves, which, as you can guess, led to a lot of mix-ups.
Yeah, early on, scientists pretty much just grouped things by how they looked on the surface. So, for example, if it had a cell wall, it got thrown into the same box. This meant tiny little bacteria were lumped in with complex algae, and mushrooms, well, mushrooms were basically considered weird plants. It was a total mess because it just ignored the huge fundamental differences in how these organisms are actually put together.
But then, right in the middle of the 20th century, a biologist came along with a truly revolutionary new system. It was a way to sort life's library not by the cover, but by the story written inside each and every cell.
The hero of our story is R.H. Whittaker. Back in 1969, he proposed a whole new way of looking at life, and it's a system that, for the most part, has really stood the test of time.
And here they are. Whittaker's big idea was to organize all life into 5 distinct kingdoms. You've got Monera, Protista, Fungi, Plantae, and Animalia. This framework finally gave biologists a logical, consistent way to categorize everything.
Okay, so let's dive into this table, because it shows you just how clever Whittaker's criteria were. Instead of just looking at the surface-level stuff, he focused on the really fundamental differences, like how complex is the cell? Is it a single-celled organism or a big multicellular body? And, this is a huge one, how does it get its food? These questions gave us a much deeper, way more accurate way to sort all of life. And right here, this is the single most important distinction in his whole system. A prokaryotic cell, like you see here, is simple. It doesn't have a nucleus to hold its DNA. Now, if you look back at that table, you'll see only one kingdom fits that description, Monera. Every other kingdom, including us, is made of more complex eukaryotic cells that do have a nucleus. This simple versus complex split, that is the first great dividing line in all of biology.
So, now that we have our basic framework, let's really narrow our focus. We're going to zoom right in on that first, most ancient, and structurally speaking, the simplest kingdom of them all, Monera.
And you know what? It's actually really straightforward. When we talk about the kingdom Monera, we are talking about one thing and one thing only: bacteria. That's it. They're the sole members of this entire group. And boy, are they survivors. Bacteria are the most abundant microorganisms on the planet. You'll find them in a handful of dirt, sure, but you'll also find them in the most extreme places you can imagine, places where pretty much nothing else can live. They are the planet's ultimate pioneers, no question.
And this brings us to a really fascinating paradox. Because while bacteria have this very simple cellular structure, their behavior and their metabolic diversity, it's just incredibly complex.
So, to really get a handle on this paradox, let's go on a quick tour of a typical bacterium. We're going to build it from the outside in, just to see how all the different parts work together.
Okay, here's our blueprint. And you'll notice right away it's way less compartmentalized than one of our own cells. So let's start our tour with those outer protective layers.
A bacterium's first line of defense is what we call its cell envelope. It's this tightly bound, 3-layered structure, and even though each layer has its own distinct job, they all work together almost like a single, flexible suit of armor.
Now that middle layer, the cell wall, is especially interesting. Based on how it's built, we can actually divide almost all bacteria into 2 major groups. You have gram-positive bacteria, which have a thick, simple wall, and then you have gram-negative bacteria with a thinner, more complex 2-layered wall. This difference is so important, it's one of the very first things microbiologists check for.
All right, now let's look at the tools on the outside. Lots of bacteria can move around using this long whip-like tail called a flagellum. They've also got these shorter hair-like things called pili, which act like tiny grappling hooks to let them attach to surfaces or even to other bacteria.
Okay, moving inside now, we find the control center. And remember, there's no nucleus. Instead, all the genetic material, this single circular chromosome, is just kind of tangled up in a region called the nucleoid.
Also floating around in the cytoplasm are thousands of ribosomes, which are the tiny little factories that build proteins. And this is where their complexity really comes into play. Besides that main chromosome, a lot of bacteria have these things called plasmids. They're these little extra rings of DNA. You can almost think of them like software plugins that can carry really useful genes like, say, resistance to an antibiotic. And get this, they can even be traded between bacteria.
All right, let's pull back out for a second. We've seen that their internal structure is pretty consistent across the board. But on the outside, bacteria come in a whole variety of distinct and recognizable shapes.
And their shapes give us a really simple way to start categorizing them. We've got spheres, which we call cocci, rods called bacilli, spirals known as spirilla, and finally these cool comma-shaped ones called vibrio.
So here's the crucial point, the main takeaway. That simple prokaryotic structure we just explored, it's like a super efficient minimalist blueprint. And that very simplicity is what allows bacteria to adapt and evolve this incredible range of metabolic and behavioral skills, letting them thrive in literally every corner of the globe.
And this all leaves us with a final thought to chew on. Bacteria have been on this planet for billions, with a B, billions of years longer than we have. So, given their immense success, what secrets of survival, adaptation, and efficiency might these tiny, ancient organisms still have to teach us?